See, your syllabus is very brief. Describe in brief the major steps involved in replication, transcription and translation. But the problem is the questions are not limited only to this thing. Questions are elaborate and you require extensive reading. And something at times comes out of the syllabus also, just like in the supply examination, which I will tell during immunology class, complement was not there, but complement was, one question was there for footnote on complement. So what I have decided that I will take 2 classes on transcription and 2 classes on translation. One will be basically dedicated to prokaryotes and another one will be on eukaryotes. The reason is very simple, that until and unless you know the basic part, which is basically involves the prokaryotic part, it will be very difficult to understand the eukaryotic part. The eukaryotic gene complex, transcription complex, replication complex, everything is very complex. So, if you have read replication, then what should I say about transcription? So, transcription, I will tell you, so what is transcription? Transcription is basically the formation of RNA from DNA. Replication is DNA to DNA, and translation is basically formation of protein from mRNA with the help of RNA and tRNA. That's it. So, why should I read at all transcription, and that too of prokaryotes and eukaryotes? See, prokaryotes we must understand because it has been extensively studied and it has been characterized, and our body is exposed to a multiple number of prokaryotes which causes disease. We want to get rid of the prokaryotes by killing them. So, until and unless we know their mechanism of growth, particularly with regard to replication, transcription and translation, and inhibition of the protein synthesized by those prokaryotes, these prokaryotes cannot be killed. And in fact, the antibiotics or chemotherapeutic agents which are used basically act on the unique system which is existing in the prokaryotes but not in the eukaryotes. So, when we want to kill bacteria or prokaryotes or protozoa or microorganisms, we have to identify unique biochemical pathway or biochemical molecule or biomolecule or enzyme which will specifically inhibit that particular substance, so that bacteria or organism or whatsoever it is will not be able to proliferate or it will be killed, and that is what is actually done, and you will be using those drugs in your clinical practice. Antibiotic, you have heard the term, their name classically, the definition of the antibiotic is basically it is derived from one particular organism which inhibits the growth of another organism, that is called an antibiotic. Now, this can be synthesized also chemically. Now, chemical agents which can also do the same thing, similar thing, are not in true sense antibiotic by definition. They can kill, they can organize, but that is altogether a different thing. So, during transcription, mRNA, tRNA and rRNA are produced. mRNA synthesis is of prime importance because it is concerned with the synthesis of protein, and as you know that every biomolecule has a lifespan. So, similarly mRNA will also have a lifespan, and in bacteria, particularly in E. coli where it has been characterized, its lifespan is only about 1 to 10 minutes time. Enzymes which are also produced by which are essentially protein in nature and they are also having a lifespan. So, in mammalian cells, they are synthesized as a precursor molecule mRNA and they undergo extensive processing. In prokaryotes, what I will be discussing shortly is basically synthesis of mRNA, but rRNA and tRNA are also produced, and the mechanism is a little bit different, which I will tell you at the end. So, if there is an error in transcription, either in the synthesis or in the modification or in the cleavage, whatever way, it will produce disease. In general, be it prokaryote, be it eukaryotes. Now, eukaryotes, one example is thalassemia, about which you know it will be called abnormal splicing. This what is splicing I will tell you, because when in the gene there will be introns and extrons. So, introns are basically intervening parts between exons which are non-coding parts. And at the end of transcription, there is splicing, means introns are removed and exons are connected, and that is called splicing. If there is an abnormal splicing also, then that will cause an abnormal protein and that will cause a disease. Antibodies also can be produced against these proteins, so they can cause autoimmune disease. There are many such things, I am not going into the detail. So, transcription in prokaryotes, you will read in books practically everywhere about E. coli, because this maximum study was done historically and correctly on E. coli, because E. coli is a simple organism, it is easy to culture, its genomes are well characterized, genes can be manipulated, and that is the reason why E. coli is studied. E. coli is, and with this, with regard to this transcription, I will tell you there are several strains of E. coli, just like human beings of multiple countries. So, E. coli K12 strain is having, all prokaryotes are having a gene of around 800 to 5000. But in prokaryotes, particularly in E. coli, since there are 4 to 88 genes, so proteins will be synthesized about the same number. One gene, one protein is usually. See, in prokaryotes, it is polycystronic, means multiple mRNA molecules are synthesized at a time. In contrast to eukaryotes, there is a single switch system, just like a one main switch control, which if you put it on, many lights in the entire campus will be on. Similarly, in prokaryotes, it is polycystronic, means at one time multiple genes from the multiple mRNA copies will be produced from a single DNA molecule, and multiple start sites are there, and that is why their transcription time is shorter. So, biomedical importance, as I told you, we have to find out some unique pathways or unique biomolecule which will inhibit specifically. Rifampicin is extensively used for tuberculosis, as I should have told you at the end. Until and unless you understand the mechanism, you will not be able to know what is beta subunit. It inhibits specifically the beta subunit, you can read it, there is no need of copying. You just try to understand what I am telling, listen carefully or attentively, follow whatever diagram I will be making, and I will be showing. Everything is written in almost in the book, so that you need not worry. Only thing is that listen, and today you finish reading at least prokaryote, so that tomorrow I can teach you eukaryotic transcription. So, actinomycin D is a drug which basically binds with the DNA. Now, if DNA is bound, it cannot be unwound. So, transcription process will not start. So, reverse transcription is a process by which a copy of DNA is produced from RNA and with the help of another enzyme called reverse transcriptase. So, zidovudin, which is used for HIV infection or AIDS, it inhibits the reverse transcriptase. So, alpha amanitin you will read in the book. Alpha amanitin is not used in human beings, but this is done for the research purposes. So, in the laboratory we want to study the activity of the enzymes or many other things, many other biochemical reactions for which some chemicals are used. Alpha amanitin is one related to prokaryote, this prokaryotic transcription. So, as you know, central dogma, there is no need DNA to RNA to protein. So, there is a similarity. If you have not read the replication, then you will not understand. The similarity of replication and transcription is same almost. Their polarity is from 5 prime to 3 prime. Okay. So, there are two chains. There is initiation, elongation and termination, and this is a multi-component complex number 1 and polymerization complex, and it adheres to the Watson and Crick model, that is base pairing rule. That is the 4th thing. So, difference is basically between the transcription and the replication is that they are in DNA you require deoxyribonucleotide, but here it is a ribonucleotide, and U is not present in DNA, and so T will be there in DNA replication or replication. Primer is required in replication, primer is not required here. Primer can be synthesized by the enzyme. In replication, the whole genome is copied to make a daughter cell, but here part of the genes are transcribed. And in DNA, there is one more important thing, that you require a proofreading, because it is giving progeny to cells. If there is any error, then that will be transmitted and that will cause production of the abnormal protein. That's why extensive proofreading mechanism is there in DNA replication, which is not present in RNA, mRNA, particularly mRNA of prokaryotes, but tRNA and rRNA undergo post-translational modification, but not proofreading, I would say. So, similarities that I have told you. So, there is a, if you want, you can memorize only thing, what is the differentiating point between replication and transcription, as I have told you. This is written in the tabular form only, nothing more than that. Remember, in replication the whole genome is replicated, and in transcription only part of the DNA molecule is transcribed as per need of the cell, not the whole one. But at times, depending on the need of the cell, multiple replication can occur in eukaryotes also. So, let me tell you a little bit about, see, there are two strands. This is 5 prime always, and this is 3 prime. This is by convention it is written, and this is 3 prime, this is 5 prime. And this is coding strand. Coding strand is represented from 5 prime to 3 prime, and this is called template strand. This starts from 3 prime end. This is called also a sense strand. This is an antisense strand. Now you have read about genetic code yesterday. What is, how the genetic codes are synthesized as per sequence of the coding strand, as per sequence of the template strand, or as per sequence of the mRNA, or as per sequence of the cDNA. Codon table, you have seen that 64 codon table. Genetic code is made from which sequence? Coding strand, template strand, mRNA strand, or something else? Template strand. Template strand is basically an RNA codon table. Remember, AUG is the starting codon. U is there. Don't get confused here in the template strand is the basis basically from which the mRNA molecules are produced, and as per sequence of the mRNA molecule, the codons are there, numbered, a triplet, AUG, TUG, U is there, T is not there. So remember that thing. So DNA codon table is also there, but classically the codon table what we refer is basically as per sequence of the mRNA molecule that you remember, and that is formed from template complex. To a template, this is called coding strand. Remember, it codes basically, but there is a conspicuous absence of T in that codon table. So this, once you copy something from, if you use this as a template, correspondingly opposite base pair will be there in the template strand, in the new strand, newly formed strand, and that will correspond with coding strand. So mRNA molecule which will be synthesized will be corresponding to the coding strand, but it will be basically from template strand, and as a rule, synthesis of DNA, synthesis of RNA takes place from 5' to 3' direction. So this is in 3' direction. So synthesis will occur in, new strand will occur from 5' to 3' direction. Okay? This part you must be clear. Always. So here in this side, if you want, so this will be also in this direction. This will be, newly strand will be 5' to 3' direction. In replication, both the process, both the strands are basically replicated, but in transcription only the template strand is transcribed. Okay? So where from this has to be started? Where from? So classically, suppose the way I am telling you, or the way you will see in the book, it are purely schematic diagram. It is not the true picture in the biological system. It is much more complex. At least at the end, one electron micrograph I will show you. After the polymerization has taken place, mRNA formation has occurred, you cannot see anything practically. Anyway, where from the DNA will start, or transcription will start, there will be one starting point, and that starting point is called transcription start point. Transcription start point, and that point is designated as +1. This is 5' end on the coding strand. So this is compared like a river, and it is called upstream on the left side and downstream on the right side. It is not up and down, because in the DNA molecule when it is present in the cell, it is really not neither in the up nor in the down position. So it is compared with almost a river metaphor is used. So anything on the left towards the 5' end is designated with minus sign. Anything which is to this side is designated as plus sign. So plus 5 means here 1, 2, 3, 4, 5. Similarly, on this side, minus 1, minus 2, minus 4. Okay. Now, how the system will recognize that where to start? So, in prokaryote, there is one sequence, there are two sequences. One is called TATA box. It is 10 base pair upstream. This is called upstream, this is called downstream. So 10 base pair minus 10 base pair upstream. There is one sequence called TATA, AT. This is called TATA box. And minus 35, there is one sequence which is called TGTT GSCA. Okay. Now, when transcription will start, something has to recognize that this particular site. So, RNA polymerase, DNA-dependent RNA polymerase, RNA polymerase. This RNA polymerase is DNA-dependent RNA polymerase, which is responsible for transcription, and fortunately there is only one RNA polymerase for prokaryotes, but in eukaryotes there are 3. In this RNAP enzyme consists of 5 subunits. Two alpha, bigger size is 2 beta, one is beta 1, and fifth one is omega. It comprises the RNAP molecule. Almost 400 kilodalton, but if you add together the molecular unit, it will be slightly more, to which one factor is called sigma factor is attached. So when everything is complete, that is called a holoenzyme. This entire structure is called holoenzyme. Basically, what is there here? It is basically alpha 2, beta prime, and omega. Okay, plus sigma factor. Sigma factor. So each unit is having one job. This sigma factor basically scans the DNA as to where the transcription site is located. Now it scans the DNA. So in E. coli basically, we are having around 4.2 into 10 to the power 6 base pair. It scans at the rate of 1000 per second and locates this particular region 35 minus 35 base pair region by scanning, and that is where the RNA actually will bind initially. That is the site, this region. A little ahead, if you go a little down, minus 10 base pair, it is exactly almost 10 base pair ahead of the transcription start site. So, omega factor, sigma factor basically scans the RNA molecule, DNA molecule, and it comes close to the site of transcription, and then it starts unwinding the DNA. So, it occurs in 6 steps. Is binding of RNAP to the promoter region. This region is called promoter region, TATA box, and this 35 base pair upstream is called promoter region. First part is binding of RNAP, this is RNA polymerase, to the promoter region and formation of a closed complex. Now, once this is formed, then the alpha subunit, there are alpha subunits are there that binds with this thing, with the DNA molecule and with the RNAP. The RNAP is there, it binds with the DNA, it starts unwinding. Beta prime subunit is endowed with the property of helicase, it unwinds. It unwinds the DNA so that the DNA molecule will unwind. And it will form some kind of bubble-like structure, just like replication bubble, there is a transcription bubble. Because this DNA has to be separated somehow, otherwise replication cannot start. So, there will be replication bubble, that is called an open complex. Means that closed structure is now a little bit open. Once it is open, then RNA beta subunit, it is endowed with the catalytic property of RNA polymerase. It binds with magnesium and then it synthesizes. Unlike DNA replication, no primer is required. Here the de novo synthesis of primer will occur. So, it will make a short stretch of RNA about 3 to 10 nucleotide length, and after these 10 nucleotides are formed, this RNAP which is attached to it needs to be cleared. This is so long attached with the promoter region, it has made an unwinding, it has unwound the DNA, some part of the synthesis has taken place, 3 to 10 base pair. Now, this needs to be released because RNAP has to work downstream. So, that RNAP molecule will be cleared. There will be RNA molecule will move there, because here when this unwinding occurs, there is temperature dependent conformational changes of the RNAP, which allows that RNA molecule to unwind. Now, once this part is complete, then there will be slight conformational changes which will release the sigma factor. Sigma factor is no longer required because the purpose job of scanning and identifying the area is over. So, sigma factor has to be released. The sigma factor will be utilized for some other RNAP molecule. Okay. So, release of sigma factor, and this RNAP molecule moves a little ahead of this thing. So this promoter region, promoter part, RNA AP is moving from this part to downstream. So promoter clearance will be there. This area will be somehow cleared, means this will be free. RNA will be freed from the promoter site. Promoter site will be there. Promoter site will be there, but RNA AP will move forward, and that is what is called promoter clearance. Now, what is the promoter clearance is that RNA AP will come here and it will start synthesizing. Now, once the unwinding occurs, there will be a torsional strain ahead. So, that is overcome by topoisomerase enzyme, both upstream and downstream. So, torsional strain will be there. So, this DNA, original DNA has to be restored back to its normal position. So, topoisomerase will be there to relieve the torsional strain ahead of the replication bubble, transcription bubble, and also on the back side, because it cannot be remain fully open. Now, this is called promoter clearance, that is formation of open complex, and the initiation part is now over. Now, once this initiation part is over, then elongation will take place. It will continue. First step here is basically formation of a dinucleotide and the formation of a phosphodiester bond. First job is first and foremost, first transcription start site, first nucleotide will be positioned, and the second nucleotide will be positioned here. Now, the synthesis is occurring here. Remember, one important part, that this is 3' end, this is 5' end. Synthesis is occurring from 5' end to 3' end, and it is the 3' end. Hydroxyl group is there. 3' end hydroxyl group is a 5' phosphate is there in the incoming nucleotide. So, bond of phosphodiester will be 3' to 5' phosphodiester bond. Synthesis is occurring from 5' to 3' direction. But since the 3' hydroxyl is free, the first ribose sugar which will attach with the phosphate bond of the second. So, the first bond, phosphodiester bond will be 3', 5', not 5' to 3'. I will show you in the picture. Now, once this short stretch of RNA is formed, then the elongation will continue. Elongation will continue so long the sequence will be there. Now, at one point of time, there will be termination signal will appear. Termination signal will be there, and that will indicate the termination of the RNA molecule. There will be termination signal. In eukaryote, there will be stop codons, but no such stop codon is there. Termination signals are there, and these areas in the template coding strand, this is a GC-rich region. GC-rich region is there. Invert repeats will be there, I will show you, and that will cause a hairpin loop-like structure, and there will be poly-A tail. Poly-A tail. So, GC-rich region and poly-A tail, GC-rich region followed by poly-A tail will be basically the termination signal. Poly-A tail here in the coding strand means basically A will be ideally replaced by T, but since this is RNA, it will be a U. So, in the mature, in the fully formed RNA transcript, you will get a GC-rich region will be there, but poly-U tail will be formed. Poly-U tail. This is basically the termination signal. Once this termination signal is reached, there are two different mechanisms. One is Rho-dependent, one is Rho-independent. I will tell you shortly. By that mechanism, RNA enzyme will be released and this newly synthesized RNA strand will be separated. This RNAP will be released. It can form another cycle of transcription. So, transcription is a cyclical process. It is not that only one transcription, one RNAP will make one RNAP enzyme will make one copy of mRNA and then it will simply die or it will be destroyed. No, it is not like that. It will be recycled. So, it will be a cyclical process. One cycle is over, it can continue another cycle and at multiple places it will reunite with the sigma factor again. There are many sigma factors for many areas of scanning. So, RNAP will be reutilized and the mRNA is now ready for use in prokaryotes. Basically, transcription and translations occur simultaneously and it occurs in the cytoplasm. Remember, transcription in prokaryotes occur in the cytoplasm, in eukaryotes it occurs in the nucleus and where it is processed and then it is sent to the cytoplasm. So, I will now tell you in briefly what an overview of what I have said. Only 15 minutes are left. I will go a little hurriedly, just follow it. Okay? You just follow it. Many things will not be written in your book. I have deliberately taken some of the photographs. So, you just go through. So, as I told you, this is template strand, coding strand, no need to tell this thing. The RNA transcript will match with the coding strand. Sequence will match. Okay? The synthesis from 5' to 3' direction, always. So, this is now, this picture will be clear. This is first strand, coding strand. You see the sequence here. You see the sequence of the transcript. This is mRNA transcript. tRNA, RNA is produced from a different site. Okay? And they are basically post-transcriptionally modified to form actually the tRNA RNA. So, this transcript which is basically an mRNA will match with this one except T. In place of T, E will be there. And now you can see the codon table, what you have seen or what you have been told, will match with this sequence of the mRNA molecule, mRNA. Okay? So, protein will be synthesized as per sequence of the thing, like AGG is forming arginine, AA is forming lysine, and then it will end with a 3' end. So, this differs between coding strand and non-coding strand, as you can understand that it is basically coding strand, basically protein coding sequence, but it will match with the mRNA. So, RNA polymerase doesn't recognize the coding strand. That is one important part. You note, RNA polymerase do not recognize coding strand, but it recognizes non-coding strand. One point I forgot to tell you about sigma factor, that sigma factor, when it is attached to the RNA polymerase, it is having a dual function. Basically, it decreases the affinity of the non-promoter region and it increases the affinity of the promoter regions and the transcription site. So, that is the function of RNAP. I forgot to tell you, it is having dual role. I will tell you shortly. So, coding sequence, as I told you, except T and U and T, already you know. So, coding and non-coding part, you see, there are two strands of DNA. Now, each strand can code for gene. So, at one point of time, the template strand from where RNA is copied, that is called a template strand. Now, the other strand is a coding strand. That coding strand can also code for some genes, because so many genes are there. It cannot be accommodated in such compact places. So, both the strands basically code for genes. Like here, you see the three genes are there, it will be clear. So, gene 1 is there in this direction. So, synthesis will occur from 5 prime to 3 prime, and gene 2, gene 3 is located in the template strand so called. So, whatever I have shown. So, gene can be present in both the strands, any of the strands. So, RNA polymerase basically require what? A template DNA strand, RNA molecule, and then RNTP, not DNTP. Ribonucleotide phosphate will be required, not deoxyribonucleotide. And then topoisomerase, as I told you, to remove the torsional strand, and the promoter region is very much essential for binding initial recognition site. So, this is what is RNA polymerase. As I told you in the picture, there are 2 alpha units, 2 beta units, 1 omega units. That is a RNA polymerase to which sigma factor is attached, and sigma factor is basically makes the holoenzyme, that is EQ. This total structure is alpha 2, beta 1, beta prime and gamma is called E, and holoenzyme is E sigma factor is holoenzyme. Now, molecular weight that I told you is nearly 400. Now, if you individually consider the weight, beta units are larger and its molecular weight is slightly more than 400. So, sigma factor, the functions, all the functions you have to remember. There is no way out, only one enzyme is there. So, alpha sigma factor is basically scanning the DNA and finding the promoter region. As I told you, it is having a dual function. It increases the affinity for decreases the affinity for the non-promoter region, but increases the affinity of the promoter regions. That you must remember. And there are 2 subunits of alpha chain. Its function is to detect and bind, assemble the RNA on the DNA molecule. So that DNA molecule will find beta prime subunit is calling unwinding, so that this replication, the transcription bubble can be formed. Beta subunit is bind, it is the catalytic unit. Remember, a MCQ can be also there. Which part actually is the catalytic, having the catalytic function? It is a beta subunit and it causes polymerization. Polymerization means basically joining of the RNA strands. Okay. So, omega subunits basically facilitates the assembly and stabilizes it. So, this is what is a schematic diagram. This is unwinding of DNA strand. It is forming a claw-like structure, so that this can bind to the DNA molecule. Okay. So, steps are 3. One is initiation, elongation and termination. Remember all these three things. So, one part is promoter. What is a transcription unit? Transcription unit is basically the promoter region, the basically signals for initiation. Signals for initiation is there for promoter region. And the initiation, elongation and termination site, the whole unit is called as transcription unit. Okay. Now, transcription unit, if you see a little bit ahead, so here you see -3 base pair ahead of the transcription start time, -35. This region, you can very well note here, this +1, there is a slightly gap. That is an untranslated region, UTR region. UTR region, although said that the transcription starts from the transcription 1 site, but in reality, this part is also translated, that UTR region is also transcribed. It is present in the mature RNA. So, I will tell you in detail later. So, if you put this as a reference point, on the left side, it is called a flanking sequence, 5 prime flanking region, and after the termination signal, this part is called 3 prime flanking region, and this must not be confused with the 5 prime untranslated region and 3 prime untranslated region. I am telling you, I should have told you tomorrow, because of the shortage of time, it is not possible. So, basically 5 prime UTR region is untranslated, means it is not translated in the protein, but it is transcribed. It is there in the transcription. So, that is what I wanted to say. Do not get confused with the 5 prime flanking region and the 5 prime untranslated region. 5 prime untranslated region is within the 5 prime flanking region. Similarly, 3' end UTR is also there. So, as I told you, the only part of the genomic DNA is transcribed at a given point of time, and it is tightly regulated, because as per need of the cell, suppose we require at the fasting stage, we require all the counter-regulated hormones to function and the enzymes to increase the blood glucose level. So, that will function when we have taken adequate amount of food. So, we want to preserve the glucose or the formation of glycogen. So, that enzymes will be induced basically. So, this is not true for the prokaryotes. So, requirements, as by this time you know that all these 5 elements will be required. So, initiation part, as I told you, it is basically the binding. First step is initiation, this is binding of the RNAP with that promoter region, and then unwinding, that is the second step, is called pre-initiation complex or open promoter complex. See, this RNAP is binding here, sigma factor is there, and it is forming a closed structure. Still, it is attached to it. Now, there is a second, this is first step. Now, there is a replication bubble, sigma factor is still attached to it. This is basically called a open complex, open promoter complex, or the transcription bubble. The TATA box, also called Pribno box, that 10 base pair ahead. So, chain initiation usually starts with the purine base, not always as a rule, but usually with a purine base, because AUG is the starting codon. So, A is basically purine, and second template directed ribonucleotide will be this dinucleotide I have told you. Now, chain initiation will occur, and once the chain initiation will occur, it will move from 5' to 3' direction. Now, phosphodiester bond, I am telling you again, which I have told you, that you see there are 2 dinucleotides. So, sugar is molecule is there, phosphate bond is here. Here, this is another phosphodiester bond. So, phosphodiester bond is from 3' to 5' direction. These I have told you in nucleotide class. So, first ester bond is there in the second nucleotide, incoming nucleotide, and the second ester bond is there in the first one. So, first one is there, the second ester bond, and this is called a phosphodiester bond. So, promoter side, promoter clearance, as I told you, once this part, the 3 to 10 base pair is synthesized, then the promoter area will be cleared. So, this is called, now the sigma factor is also released, as I told you. Now, this promoter region is cleared, it is moving ahead a little bit after this replication bubble. Now, chain elongation will start, and it will go on adding at the 3' end of the transcription sequence. Elongation will start as per sequence of the RNA molecule or DNA molecule. So, 10 sequence will be there, chain elongation will take place, and RNA AP will be still attached, only thing is that sigma factor was released. Now, this is what I told you. So far, we have told so many schematic things, but after the transcription has taken place, this will be the picture in the electron micrograph. So, from that you cannot elucidate anything. What I have told you so nicely, schematically, and it is written in the book. In reality, from here we have to basically go at the molecular level. What is the actual structure? You can see the transcription start side. I have told you so clearly on the blackboard, transcription bubble, everything. Now, you see this is the transcription end side, this is the transcription end side, and this is how this transcription actually has occurred. This is the picture. So, it is not that simple as we see, and in reality it is still difficult in the live situation. So, chain termination will occur and RNAP will be released, and this will be by 2 different mechanisms. One is Rho-dependent and Rho-independent. Normally what happens, there is Rho-independent means normally, as I told you, when the termination signal will be there, GC region rich will be there. So, GC region rich will make an hairpin like structure. Okay. So, that will form a poly U tail. So, that will automatically, that area will be recognized by the RNAP and it will clip that RNA molecule, mRNA molecule, and it will be released. So that is called an Rho factor independent, because there are two mechanisms spontaneously. So RNAP molecule is having endowed with 5 different functions. First and foremost, it binds with the promoter region. It is having a primase activity. You will say first the initial primer binding also will occur. It will cause unwinding, it will cause polymerization, and it will also terminate and remove the RNA molecule from the DNA strand. So it is having a multiple function. All in one is basically present in the RNA polymerase. It is not so in DNA polymerase molecule, as you will see it tomorrow. So it will form a hairpin-like loop structure, and this will facilitate automatic removal of mRNA molecule. This is Rho factor independent transmission. See, you see here, this hairpin loop-like structure is there. So RNA polymerase will not be basically able to move further. So that will cause a signal and it will be terminated. So Rho factor dependent termination. So when this factor will not operate, then only we require a Rho factor. Rho factor is basically a protein that will bind to a Rho utilization site. I will tell you in this picture it will be clear. So this is the growing chain. So Rho utilization site will be there. Rho factor is basically a protein in nature. It is endowed with 2 activities. One is ATPase activity. So ATP will be required here, and it is also a helicase activity. It will unwind. So Rho factor binds with the Rho utilization site. RUT means Rho utilization site. It will bind with the RUT region, and then it is a motor protein. So it will move like a spirally, and it will reach to this site, as I can show you. So this is the Rho protein, Rho factor. It will bind with the RUT site, and then it will move from here. It will move. You see, it has moved here. Now it has reached to this place. Once it has reached to this place, it will automatically clip with requirement of 1 ATP. So energy will be required here. That is called Rho-dependent termination of RNA molecule. Okay. So this is what is in short synthesis elongation. So post-transcriptional modification, there is no post-transcriptional modification for mRNA, but for tRNA and RNA undergo extensive post-transcriptional processing. Regulation must be there, because without regulation, unregulated amount of mRNA will be produced. It is not so. This is a very difficult process, tedious process, and nature will not spend so much of energy and time for doing this thing. Only thing is that its control degradation will be under control to some extent by the regulatory genes which are present within the mainly within the 5' franking region, and also some in the 3' franking regions. So transcription is basically a cyclical process. Once a cycle is over, it will be ready for the. So as you see RNA binding and close complex formation. I am repeating once again so that you remember at least this cycle. You read once today. It is not that difficult. Tomorrow it will be easier for you to understand. So RNAP binding formation, then that is a closed formation, then open formation, then chain initiation, chain elongation, chain termination, and it is ready for another cycle. So this is called a transcription cycle. This transcription process is basically called a transcription cycle. So স্যার has come, দেখো তো স্যার চলে এসছে, ঠিক আছে, তাহলে কালকে এটা পড়াবো, একবার একটুখানি পড়ে নেবো আজকে, ওকে? তাহলে এই প্রোক্যারিওটিক আমি ভাবলাম যে একটু খানিকটা কভার করে দিতে পারলে সুবিধা হতো, কালকে ক্লাসটা তাহলে অনেক লেন্থি হয়ে যাবে। একটু ডিফারেন্সিয়েটিং পয়েন্টগুলো বুঝতে হবে। Only thing is that because this can come as a question also.